So this group is unique in that sense that its common feature is that uh, it includes some bacteria which predates on the other bacteria. Right, so far you have discussed so many nutritional behaviors in bacteria. It may be photosynthetic, it may be heterotrophic, it may be symbiotic, it may be parasitic, but predation is something very unique that you have not heard or discussed before. So these members, some of the members of this Deltaproteobacteria, it includes those bacteria that predates on other bacteria. They are the predator on the other bacteria. So, and also they contribute to the sulfur cycle, right? So since I have already discussed that, we will discuss only those members which have some commercial or clinical or any environmental significance, right? Or any, say, any distinctive feature. If any group or if any genus is having any distinctive features, only those will be discussed in these lectures, right? So these two properties, they are displaying group properties, right? And the remarkable feature of this group, right? So the first that we'll discuss is relevant to predation, bacteria predating the bacteria, and one of the member is the Bdellovibrio. B is silent here, right? So it's Bdellovibrio, and as you can see in the picture that it is shown in the yellow color, and in blue is representing the host cell or prey, you can see, and it is also gram negative because since you are discussing the proteobacteria, which is a group of gram negative organisms, but interestingly it attacks other gram negative organisms. And how it attacks the other gram negative organisms, it penetrates the outer layer of the other gram negative bacteria, but it doesn't invade the cytoplasm, rather it maintains itself in the periplasm of the gram negative organism. So the cells, where whenever they attack the other gram negative organism after penetrating into the periplasm of the prey, or you can say the host, so they elongates into a tight spiral. अपने आप को ये बहुत ज़्यादा elongate कर लेते हैं, and then they undergo fragmentation into several individual flagellated cells, and then the host cell, it lies and then it releases the newly formed Bdellovibrio cells. This is how it reproduces itself. So it's something very unique and distinctive that I thought that it must be discussed in some detail. The second thing that I have discussed previously, that the two important feature is that either they are predating the other bacteria, or the second important thing is they are helpful in maintaining the sulfur cycle, right? So in the previous group you discussed a member, Thiobacillus, which was responsible for the oxidation of sulfur to sulfates or to elemental sulfur, but this group is responsible for sulfur reduction, means it can transform the sulfates into its reduced form, that is hydrogen sulfide, H2S, right? So they are transforming sulfates, available sulfates and elemental sulfur into reduced form, that is hydrogen sulfide. So wherever hydrogen sulfide is generated, so generally it is the environment which is anaerobic. So usually they are present in the anaerobic sediments, And the other thing is the intestinal tracts of humans and animals, which is another anaerobic environment, and it is the best studied sulfur reducing genus, and that's why its name is indicates Desulfovibrio. So they use oxidized form of sulfur such as sulfates and elemental sulfur rather than oxygen as electron acceptor, right? So as a uh, with a, you can say in replacement of oxygen they use sulfates, and they use elemental sulfur, and they transform it into H2S, and that H2S is used in replacement of H2O. So that's how they are maintaining their electron transport chain, and they generate their energy using in the anaerobic environment. Right, so the product of this reduction I told you is hydrogen sulfide, and whenever hydrogen sulfide is produced, it reacts with the iron and it forms ferrous sulfate or iron sulfate, and it is responsible for black color of many sediments in which these desulfovibrio are present, right? So blackening of the sediments, they are because of this reaction by these microorganisms. Myxococcus, it's another very interesting genus grouped in the Deltaproteobacteria, and Myxococcus is known by its mucus-like secretion. Basically, this form is very unique, and you will be amazed to see that how beautiful they display themselves. So, and they are visible. So, how they are visible? Because they do not mostly they are in form of aggregates, right? So they are similar in their morphology, and like the slime molds you probably discussed in your intermediate classes, right? So Myxococcus is known for its ability to form the mucus-like secretions, right? And these secretions, they help these bacterial aggregates to glide and glide together. So basically what they do, they aggregate themselves, and when multiple cells they aggregate together, they form a large a visible mass, you can say, and together when they are in the aggregated form, they release mucus like secretion, which helps this aggregate to move by gliding, and they can be detected by their slime like trail, right? They leave a slime trail behind, and you can notice the path of the Myxococcus. So how they move, I told you that they move through gliding, and gliding is facilitated by the secretions, nasal mucus like secretions, and the common or well representative species, they include Myxococcus xanthus and Myxococcus fulvus. So basically uh, when they move, so they actually engulf the uh, smaller sized bacteria they encounter, because they are now in the form of a large mass, right? So they encounter the other bacteria and they engulf them, and they lyse those uh, bacterial cells enzymatically, and then they digest, and that digested or you can say the nutrients, they are distributed in the whole uh, aggregate, or you can say in the whole bacterial community, I can say. So they form like a uh, they share their metabolic activities together, and they are centrally aggregated at one place, and they form like a body and a mass, which is a characteristic feature of this genus, which is known as Myxococcus, right? So its life cycle is somehow similar with that of slime mold. So basically there are some triggers, right, that indicate that induce this formation of aggregated body, I can say. So this induction, or you can say this could be because of the low number of nutrients available or scarcity of nutrients available. So what they do, they tend to aggregate themselves, right? So when they aggregate themselves, so what happens, their aggregation, it leads to the formation of a mass, and later on, um, from that aggregated mass, what happens, and there that mass or some of the portion of that mass is differentiated into these structures, and they form fruiting bodies, which are also known as sporangioles. And in these fruiting bodies, what happens, definitely this is in the slide of life cycle, so definitely it involves the mode of reproduction. So this is how they reproduce, and this is how they form aggregates, and they not every time they exist in the aggregated form, but it is a part of the cycle, right? That once they are aggregated, so they later on they form a differentiating body, which is known as a fruiting body as well, and this contains myxospores. And these myxospores, each of the myxospore is then germinated into a new individual vegetative cell, right? So basically from this to this, there are different you can say triggers, या ये जो aggregate करते हैं उसके लिए there are different triggers. But once the situations are favorable, only then these myxospores they germinate themselves, and when they germinate, they each of the myxospore develops into a vegetative growth cycle, right? So they remain separated, or they remain individually once the things they are uh, normal, or you can say that nutrition is available, they are provided with nutritional requirement is fulfilled, and so they do not have the requirement of aggregation at that time, right? But under certain conditions, I discussed that it may be drought, it may be nutrient scarcity. So what happens, these vegetative cells, they swarm to central locations, and they move together, they move towards a single place, you can say, and after reaching there, they form aggregates, right? And when they form aggregates, so I told you that there is a mass, and with that mass is known as the mound, or the process is known as the mounding. And it is a sort of early fruiting body, or you can say the non-differentiating fruiting body, right? Which later on then develops into a differentiating fruiting bodies, right? And from this fruiting body again, myxospores they start to form, right? So this is the whole life cycle of the Myxococcus, and which is very unique in that sense that this type of life cycle is not usually found in the bacterial domain,